Hello, and welcome to the Axon Instruments webinar series. My name is Jeff Weber, and I'm the product manager for electrophysiology at Molecular Devices. I'm very excited today to introduce Dr. Balaji Krishnan from University of Texas Medical Branch at Galveston. Dr. Krishnan will be discussing using patch clamp electrophysiological studies to accelerate mechanistic studies and synaptic transmission and reception. And now let's jump in with an introduction to our speaker, Dr. Balaji Krishnan. Dr. Krishnan is an assistant professor in the Department of Neurology at the Mitchell Center for Neurodegenerative Diseases, which is part of the University of Texas Medical Branch in Galveston, Texas. Dr. Krishnan is interested in memory mechanisms and is focusing currently on synaptic dysfunction as the basis for memory deficits. The goal of these studies is to identify mechanisms that are amenable to therapeutics and therefore applicable for preventing memory loss. Dr. Krishnan received his bachelor's in microbiology and biochemistry and master's degree in molecular biology and physics from the University of Mumbai, and received his PhD from the University of Texas studying the neurogenetics of circadian rhythms in olfactory responses. And now I'll turn it over to Dr. Krishnan.
Thank you, Jeff. Thank you for giving, that was a very nice introduction. Thank you so much. I would like to thank Molecular Devices and everyone that I have been interacting with. They have been amazing, and I'm very happy to present this webinar, which is going to describe the electrophysiological studies and how to use it as a mechanistic tool to look further into reception and transmission. So the structure of my talk is going to be as follows. So first I'm going to introduce you to peripheral neurotransmission. So this is talking about sensory systems using the fruit fly Drosophila melanogaster. So this is going to be the invertebrate system. This will be followed by how these studies were then taken over into looking at central mechanisms, which is the peripheral circadian rhythms in the fruit fly and how it influences signaling in the center. And this will be followed by looking at how you can use electrophysiology, especially field electrophysiology, which studies synaptic connections in the vertebrate system for studying long-term memory. And what I have done so far in trying to discover a particular protein that is important for long-term memory expression, both in appetitive, which is memories that you like, and aversive, which is memories that you don't like. So we start off with the humble fruit fly, the Drosophila melanogaster. I'm sure most of you are very familiar with this fly, which hovers around anything that is ripened or anything that is around. It is not as nagging or painful as the housefly, but it is painful nonetheless. I've heard many people complain about it. Anywho, this is the male fly, which is smaller than the female fly, and this is the head, the thorax, the abdomen, and the wings. These are the distinctive features. The life cycle of the fly consists of 10 days. It is made up of, it is completed in 10 days. So you have the embryo, followed by the first, second, and third instar larvae, which are 1 day each, a pre-pupal stage, the pupal stage, and then finally the fly. So within this one organism, you have two different stages. And what I'm going to be focusing on today is the adult stage. So what is the specific features of Drosophila that make it very useful? First of all, evolutionarily, it is midway between bacteria and humans. It has a well-defined and highly manipulable genetics. So over 100 years of history in this fruit fly has allowed us so many different manipulations that it is amenable to so many complex behaviors, which is not possible in the worm, and sometimes it's difficult to translate into genetics in the mouse and higher vertebrates. So this becomes an ideal midpoint to look at all of this. So in my talk, I'm going to highlight how you can use not just one, but different model organisms in order to study the question, which is going to advance science. So you're not restricted to one particular organism, but I would like to start off with how Drosophila was the one that I found useful, but then had to move on to look at the vertebrate systems. Nevertheless, coming back to the fruit fly, there's about 60% of the genes that are conserved between the two species. 75% of known human diseases have a recognizable match in the genome of the fruit flies. That makes it really important. 50% of the fly protein synthesis have mammalian homologs, so it becomes easy to study complex human genes which have more pleiotropic function than the fly gene. So we can go back to the fly, look at it, and then advance it to the humans. There is, of course, a lot more online database available, so you can pick out a human disease, find whether it has a homolog in flies, find out what kind of research is going on. So this makes it very useful for even the common public. And recently, Drosophila has become a model organism for choice as a genetic model for many of the complex human diseases, including the one that I am currently interested in, which includes Alzheimer's disease, and also, in addition to that, studying aging, oxidative stress, immunity, diabetes, cancer, as well as drug abuse. So it's a very versatile organism. I am going to focus today on the head of the fly, and this is a picture micrograph of the head. It is a scanning electron microscope. What I would like to highlight is the eyes, which are located on the sides. These are compound eyes, and the head structure over here is as follows. Electroantennograms, the technique that I'm going to talk about, is a extracuticular recording, which means you record from a population of neurons that are located on the sense organ. So in this case, this is the antenna. So hence the name electroantennogram. It was first described in the moth, and it is an electrical response to sensory stimuli. So the sensory stimuli in this case is basically different kinds of odor and different concentrations of odorants. And when I say odorants, these are volatile chemicals like ethyl acetate or alcohol, things that evaporate. So these are sensed by these two particular sets of organs that are labeled here, called the antennae and the maxillary palps. And for today's study, I'll be focusing on the antennae because this is the major sense organ that is receiving information about ethyl alcohol, isomyl alcohol, which are fruit odors, as well as repulsive odors, which are emanated from predators of the Drosophila, like reptiles and lizards and so on. So, how do we record extracuticular responses? Well, this is where I'm going to demonstrate first how we did it. So you take the fly. The best part of the organism is you can take the whole fly, have it alive, and then you immobilize it using this compound called as myristic acid, which is basically wax. It melts at 8.5 degrees Celsius. So you can melt it quickly and use it to immobilize the fly when it is alive. So right now in this position, which is through the hole in a plexiglass sheet, you basically have it tethered and only the head with the antenna is suspended. So when you look at it from the top, this is how it looks like. You have an array of 6 to 8 of these flies which are tethered. Here is the airstream which will deliver the odor, and these are the 3 electrodes that are used in this particular preparation. Your fly head, the hook which is used to lift up the antenna, the ground electrode which impales into the tip of the antenna, and the recording electrode which is placed on the side. So here is a cartoon representation of how it looks like. The ground electrode, the recording electrode which goes to the amplifier, and this is the head of the fly. Now, you have a structure called as the olfactometer, to which air is supplied. It is a set of tubes with different odorants or different concentrations of odorants. When they are delivered onto the fly antenna gives out an electrical response, which is collected by the recording electrode, amplified. The audio monitor allows you to make sure that the connection is complete, and that is again tethered to the amplifier and the digital data acquisition unit, and that goes to the oscilloscope. So here is a representation of 2 of the units that I've used for this particular preparation, and we'll come to it again. This is the multi-clamp 700B, which is capable of recording up to 2 or 4 at a time, and this is the Digidata 1550B, which has the hum silencer and allows acquisition of the data. The others I'll come to down the line. So how do the responses look like? So I'm showing you the responses that we observed in female flies. So here is the baseline trace, okay? And when you supply or give a puff of odor, which is interrupting the airstream. This is at a concentration of 10 raised to minus 10. So this is basically serial dilutions that I'm showing for a compound called as ethyl acetate. This volatile compound gives a small response, which is seen as a downward deflection, which increases with increasing concentration. As you can see here, the amplitude keeps increasing. So you can use the measure of this amplitude, which is plotted here in this graph for 24 flies, as a readout for this particular response. So using this, we characterize about 48 different mutants. And I'm going to give you the power of this particular technique in able to tease out mutants just by looking at the functional profile. So here, showing you along the y-axis is the response in millivolts, which is what you saw as the amplitude over here. As you can see, the millivolts keep on increasing. So this is at different concentrations for one particular compound, ethyl acetate. The second one is for isomal acetate, and the third one is for N-butyl acetate. The white hollow circles represent the wild type response. CS is the wild type flight. So you can see that there is a profile of response. As you can see, there is a difference between this compound, the N-butyl acetate, versus isomal acetate or ethyl acetate. That is because N-butyl acetate is not as volatile as isomal or ethyl acetate. All A402 is a P element mutation. This was generated by a P element. Excuse me, just a second. So this was generated by a P element mutation called as OLF402. So this insertion, just a second, just give me 1 second. My apologies. So this P element mutation was inserted into the genome of the fly in a search for different receptor mutations. So what you see over here is a receptor mutation that increases the response. So you can see here in black, it increases the response to ethyl acetate much more than the wild type, which is a unique phenomenon because usually you expect that if you mutate a receptor, you're going to see a lesser response. So this is an example of how this strong, powerful tool can be used to assay not only decrements but also increases. As you can see, the specificity, it is not showing up in the other two olfactory odorants that we used. So here is another example. Here you see a sexually dimorphic response. So here shown along the right-hand side panels are the female flies. Again, I'll point out that white is showing the wild type. You see a particular response to ethyl acetate. These are two different regions of the antenna, a medial region and an anterior region. So again, this is the advantage of the field recording. You can use different regions of the antenna to look at different regional neuronal responses. So as you can see from the black circles over here, the OLF-A186, which is another P element insertion, does not show much change in the females. However, in the males, it is significantly decreased compared to the wild type response. So here we mapped out another olfactory receptor mutation, potential one, which is affecting one sex but not the other. Here is a third one. This third one is even more interesting because what you would expect is if, depending on the specificity, if there is an odorant that affects a mildly volatile odorant, then it is going to show a similar profile with all the others. That was not what we observed with all A534. This particular mutation, cause or insertion as you will, decrease the response to just N-butyl acetate, not isomal or ethyl acetate. So this is just showing you a small collection of mutants that allow you to map different odorant receptors. Now, giving you a summary of this sensory reception studies, you can take these three distinct kinds, one that is affected with volatile, a specific volatile one, the other that has a sexually dimorphic profile, and the third that is responsive to less volatile odorant and do further behavior as adults, which I did, and saw that this profile matched up. And then you can look at biochemistry, map out where this gene is, and find out what are the characteristics of this receptor, which we found out to be a 7-trans membrane, which is giving rise to different responses downstream. So this started off a cascade of different studies, which I was very happy to complete as a part of my master's. Once I finished these studies, I was very eager to apply this to something that is more fundamental. And this is where I moved over to do my PhD at University of Houston here in Texas. So to give you an overview, I wanted to look at how sensory reception can affect circadian rhythms. Circadian rhythms are defined as a particular central clock that is present in the body, especially in the brain of vertebrates as well as invertebrates. So these are a set of neurons that receive input from the environment. And since our strongest environmental input that has a regular cycle is the sun, the input resets the clock every time the sun shows up, which gives rise to humoral or neuronal signals that will then regulate a bunch of rhythms. For example, the locomotor activity rhythm in the fruit fly is governed by the rising and setting of the sun. So if you put the Drosophila into a small tube and monitor its activity in the presence of this sunlight, which you can mimic inside a room by shining artificial light, you will see that the clock resets itself at 2 different times of the day, one during the dawn and one during the evening or twilight hours. Now, the question that I wanted to ask was, if you look at, say, the antenna, for example, which senses odor, can these odors or anything that affects the odor, like temperature, can this run the clock instead of the sun? So that is why the dotted arrow. And when that sets a rhythm in the sensory system, can that then control the behavior of the organism? To ask this question, the first thing I had to do was to discover whether there is a rhythm in the sensory responses. This is where the electroantennogram technique came very handy. So here I show you the first publication that came out, where what I did was I took one concentration of ethyl acetate, I took the millivolt response and saw whether it changes as a function of time of day. This bar above just shows you that since I had to do the experiments during the light, for the first day, the clock can maintain the rhythm. But if you keep the lights on for a very long time, then the rhythm goes away. So what I did was I did electroantennogram recordings and looked at 1 hour, 5 hours, 7 hours, 13 hours, 17 hours, and then 21 hours. And those are the shown in the rectangular blocks are the wild type responses. You can see basically the EEG responses are flat, and then in the night, in the subjective night, the response increases. So why would you have a response increasing in the middle of the night? Is this a true response? Well, Drosophila offers the best tool to do this. So we selected 2 circadian mutants, period and timeless, which have deficiencies in the circadian clock. And when we looked at those guys, look over here, these 2 with the hollow circles and the fair circles, there is basically no change. You don't see this rise. Not only that, if you keep this rhythm on for 3 days, so if you keep the flies in light for over 72 hours, then after the 3rd day, you basically abolish this rhythm. This is very encouraging. And when we thought about this process, it came to us that it was very obvious. The fly is an organism that requires vision for all its activities. That is why it is active in the dawn hours and in the evening hours. In the middle of the day, because of dehydration, it does not have much activity. As a result, it is depending on its vision for its activities. However, at night, when the predators when it is taking rest and the predators like reptiles are present, it needs a way to sense them where it cannot see them. And hence, it allows us to provide a hypothesis that at that time the sensory sensitivity is increased such that the fly can detect and respond to it. So now, encouraged by these results, I wanted to find out the second part of the question. Okay, is this rhythm controlled in the antenna itself? In other words, is there an oscillator present in the antenna? And I got some evidence from the vertebrate field on this regard. If you recollect, I mentioned to you that Drosophila is midway between evolution of the bacteria to the humans. So this turned out to be very useful for me because I looked at the mammalian system and found that cryptochrome, which are basically flavin-terrin proteins, these are involved in the central coarse clock function in the mice, but they didn't have a role in the Drosophila central oscillator. So I took this mutant and asked the question, if I change the temperature, okay, if I give it high temperature and low temperature as the zeit giver or the time giver. So like I mentioned, environment can entrain the clock. So this entrainment can be done even with temperature. So if you look at it, you do see a rhythm in the wild type, which is shown in this black trace, which is completely abolished in the cry mutant. So while Per and Tim showed a rhythm, cry knocked it off. This was very encouraging because this suggested that this peripheral rhythm may be under the control of cryptochrome. So shown here are what happens if you basically have it in constant temperature, either high or low. You get basically the rhythm present, but it's abolished in the cryptochrome mutant. So with this, I will summarize what we obtained and give you the implications of this study. So using electroantennograms, I hope I have been able to demonstrate how you can use it to study circadian rhythms, a central function, and mapping this genetically by using the powerful tool in Drosophila of genetics. Okay. Now, this is the first proof that a circadian oscillator is present in the periphery. And this article of mine, both articles have been cited several times because of the fact that you can use this and translate it into humans. So, for example, you can use smell, you can use sound, you can use other entrainment agents to give it to people who do not experience sun. For example, in climates with higher altitudes like Norway and so on, you do not have the sun at regular intervals. So the circadian rhythms in the humans gets messed up, which gives rise to seasonal affective disorders. Now you can use things like smell, things like taste, things like touch, all of these as sensory agents to train the clock such that you do not suffer from these mood disorders. When I discovered the implications of my study, it blew my mind, and I wanted to see what other things we can discover in vertebrates. And this is why my attention shifted to vertebrate electrophysiological studies. And I was naturally interested in the central mechanism of learning and memory because smell, circadian rhythms, everything has an influence on learning and memory. So what I'm going to show you are the studies that I did at University of Texas Medical Branch as a postdoctoral fellow, discovering a key molecule that is important for aversive memory. And this occurs in the emotional brain called as the amygdala, and I use rats to do these fear condition studies. I took the studies further to look at appetitive memories, which are memories that you like, again controlled by the amygdala, and for this I use cocaine conditioned memory. And the model organism was a rat. So this allowed me to discover a new role for phospholipase D. And I won't go much into the details of the signaling mechanism, but suffice to tell you that phospholipase D or PLD is a phospholipase similar to phospholipase C, but it is only recruited for long-term memory mechanism and recall, which is what I discovered using my electrophysiological studies and others. So, to align you to the topic. So if a rat were facing you from the screen and you took sections of his brain, this is basically how it will look like. So this is a coronal rat brain slice. And in this coronal rat brain slice, I want you to focus in this particular area over here in the red boxes. Okay? This is the region called as the amygdala. Amygdala stands for basically almond-shaped, which is what it is in humans. Over here is the typically recognized structures of the hippocampus. The amygdala and the hippocampus form the associative memory parts of the brain. In other words, when you form a memory, these two structures are involved. The amygdala contributes to the cue, which is the sound, taste, smell. Remember, I'm interested in sensory systems, so that is what I was driven by. So all of this information is collected. So if you like something, then that information is stored as memory in the amygdala. The hippocampus gives you the context. So, oh, this is where the tree was. This is where the place was. So together, the amygdala and the hippocampus are the main structures that give rise to all the things that we see in our learning and memory. So I gravitated towards the amygdala because of the importance in sensory function. So to give you an idea, any kind of information goes to the thalamus, which is the central structure, and then it's routed to the brain to get the memory. However, there is a separate connection, which is called the cortical afferents, that always parallelly goes to the lateral amygdala or to the basolateral amygdala. These are nuclei of the amygdala that are important, and that information is conveyed to the central amygdala. So these synaptic pathways between these three circuits are very important. I'll come to it again so you won't miss out on what I'm trying to say. Then the central amygdala is responsible for the behaviors that you see. So for example, if your eye notices a snake, that information is quickly conveyed to the lateral amygdala. The lateral amygdala says to the central amygdala, oh my God, it's a snake, run. So that information is then conveyed by the central amygdala to the rest of the body, so it translates into behavior. That was just a simplification, but that's to give you an idea of how the integration occurs. So here are the amygdala synaptic pathways. So again, this is a slice of the brain. So from now on, the study that I show you, we don't use the whole organism, but we use brain slices. And it's beyond the scope of this particular webinar, but that's a webinar by itself on how you can make different brain slices. Suffice to say, this is a coronal brain slice. And here you can see the lateral amygdala, the basolateral amygdala, and the central amygdala. These are three different nuclei with three distinct functions. Okay? And all of these cross things that you see are the fibers. So this is a highly dense, non-homogeneous structure compared to the hippocampus, which most people are used to, which is a regular structure. So what happens? Sound, when you pair it to the foot shop, and this is for fear conditioning, the information is conveyed to the auditory system and the somatosensory system. Foot shop to the somatosensory and sound through the auditory to the lateral amygdala of the lateral nucleus of the amygdala. This is then conveyed to the central and maybe to the basolateral through interstitial neurons that are present over here, which are inhibitory, to then give you an emotional response from the central amygdala. Okay? This is the structure, or this is the hypothesis proposed by many eminent scientists that then Ledoux presented in this nice cartoon format. When I joined the lab of Dr. Patricia Schmidt-Gallagher at UTMB, who is one of the pioneers of amygdala electrophysiology, here I'm going to show you basically the central amygdala and the basolateral and lateral amygdala as these cartoon structures. What was known was the NMDA currents and the receptor proteins. So again, now looking for the signaling pathway, we knew that glutamate is important and it is reduced during maintenance of fear memory. So here is where the difference comes. Dr. Gallagher, Schmidt-Gallagher, looked at animals after they had learned the fear memory to see what happens in the circuit. And what she discovered was there was a change, a complete change in the glutamate signaling, okay, where NMDA reception or NMDA neurotransmission was reduced. Not only that, this connection between the lateral and the basolateral amygdala, which is going to tell information of fear, to all the cues, that was also not invoked. So in other words, information came from the thalamus to the lateral, went to the central, was not routed to the basolateral. Whatever information was required from the basolateral, that was separate. It involved NMDA, it involved L-type voltage-gated calcium channels, and these were unique from what you saw here. So it became important for me to map this particular circuitry and see which one is important for fear and which one is not, which one is important for appetitive memories. So for this, we had to use fear memory. We had to use memory in which the animals either liked or disliked or showed a preference for cooking. So these kind of behaviors I'll not be able to go into, but I'll tell you the gist behind it. So here again, I will show you how we took the responses. So in this slide, you place a stimulating electrode, and in this case, you can do stimulation using electrical currents or chemical compounds, and I'll talk about both of them. So to begin with, you place a stimulating electrode in the area, in the presynaptic area, and you record in the postsynaptic area. So this is the thalamic to the LA synaptic circuit. So here is your field excitatory postsynaptic potential, or FEPSP. When you look at this FEPSP, you of course see the stimulation artifact, which comes from the stimulating current, and then the baseline shifts downwards because a whole population of neurons are responding, and since you have just placed the recording electrode on top of the lateral amygdala, you are just getting the field potential, the local change in the current, which is showing as, it's not a depolarization, but a change in the amplitude. The more consistent part is the change in the slope. In other words, sometimes you can see that the amplitude before stimulation would be, say, somewhere over here, and then it shifts and becomes a sharper, steeper slope, okay? And that is what is called as the slope measurement. So all of the studies I will show you, I'm looking at the slope, which is the millivolts per millisecond. Here is where the complete utility of this particular rig comes into handy. So here is your stimulating unit, which is basically the anti, which is connected to a isolation unit. However, the beauty of this is using ClampX, the protocols in ClampX, you can program this particular setup to be connected to your 1550D, the digidata acquisition, and then the amplifier controls, the digital interface controls both the stimulation and the recording. And this makes the whole system very powerful and unique. More importantly, I found that it was very versatile, and you could input a lot of different stimulating strategies because it was under the control of ClampX. So now, going back to the mechanism, using this powerful technique, what we did was we stimulated in this area and recorded in this area. Stimulation was in the thalamic, recording was in the lateral. And what you see here is the percentage of baseline FEPSP slope. So what you saw there as millivolts per millisecond was taken. We did a 10-minute baseline, and then after that 10-minute baseline, we looked at what happens after you give the current. So shown along the y-axis, you have the frequency measured in hertz. So 1 hertz implies that one current stimulus was given per 60 seconds, 10 is 10 stimuli, and 100 is 100 bursts. So these are called as 10 hertz and 100 hertz, and TVS is a special one, which I'll not be talking about too much today. Okay, so what do you see here? If you take the naive animal shown here in the black circles, you can see that at 1 hertz, you have absolutely no change. Basically, the system is not responding. The synapse is not getting potentiated. However, when you increase it to 10 hertz, you see a slight potentiation, about 120%. Okay. Then when you increase it to 100, you see a 160% increase in the response. In other words, the natural tendency of the synapse is to get potentiated. So LTP is the cellular basis of memory. This is by far the best possible way for us, in addition to the in vivo, to map anything that is happening in the brain, okay, after taking slices of the brain and recording from it. So that is what I'm showing you here, the power of this technique in being able to respond to currents in this manner. So using this, what Brad Schroeder, a student in Dr. Gallagher's lab, showed was that fear memories induce a switch in the stimulus response. What does that mean? If you condition the animals to fear, then you see the opposite trend. You can see this hollow circle, which basically drops down to almost zero. So it shows the opposite profile. The correct control is not the naive animal, but the unpaired animal. What is the unpaired animal? Like I told you, amygdala is important for associative memory, which means you have to give the shock and the sound in very close proximity to one another. They cannot be very far off. If you make them far off, in other words, this group of animals received both the same amount of shocks and the sound, but they were not paired. They were unpaired, hence UP. You see that it basically behaves very similar to the naive animals. So this shows you that if you potentiate or if you develop fear memory in the circuit, because it stores memory, the synaptic connections are already changed. Now my question was to find out how we can look at this. So this is basically just showing you that if you look at, I want to use this as a template to show you all the other recordings that is happening. So you will see that along the y-axis you have time in minutes. You have a baseline response where there is no difference between any of the three experimental groups that I'm using. Then after you give the low frequency stimulation, which is basically 1 hertz, you will see that the naive and unpaired do not show any potentiation, whereas the fear conditioned group is showing you potentiation at around 140%. Okay? So using this, I went back to the thalamo-LA synaptic circuit in the fear memory and looked at what happens if you give a 100 hertz stimulus. If you give a 100 hertz stimulus, the baseline is fine between the paired and the unpaired animals. The LTP, which is the long-term potentiation, seen after an hour of giving the high-frequency stimulation, is not distinguishable between the paired and unpaired. However, the post-tetanic potentiation definitely shows a change. So the first 10 minutes, which shows more of the presynaptic profile, there is a change. And I mapped it using this post-tetanic potentiation protocol and studying the parameters of this to show that if you used a drug called PCCG13, which is an inhibitor of phospholipase D, especially the mGlu-driven phospholipase D. So here is where we come to metabotropic glutamate receptors. When they signal phospholipase D as a second messenger, then you get this response. How do I know this? Because if I block it at this stage, then I do not see any change in the post-tetanic potentiation. So this tells you how you can use high-frequency stimulation as a way, or electrical stimulation as a way, to map out circuits. Phospholipase D was discovered as a part of a two-dimensional gel electrophoresis, where we saw that this was particularly increased. So I started studying the mechanisms around it, and I'll explain some more as we go down the line. But to summarize the fear conditioning studies, aversive memories involve phospholipase D as a long-term signaling molecule, especially for the expression of memory. Based on post-tetanic potentiation studies, a presynaptic control of vesicular traffic may contribute among other postsynaptic events. What do I mean by this? Phospholipase D is a phospholipase, which means basically it changes the architecture of the synaptic membranes. So it can basically control the synaptic membranes such that more or less vesicles can be released. And if the levels are increased, levels of phospholipase D increase, then this means the whole circuitry is changed. So this increase in phospholipase D levels could be a great mechanism at the synapse that is controlling long-term memory. That is what I indicate by that statement. Of course, field electrophysiology studies are very important for mapping synaptic circuit between brain regions. So you can think about this. I have used patch clamp, but patch clamp tells us information about a particular cell or between two particular cells in a synaptic circuit. If you want to look at whole synaptic area, then field electrophysiology becomes an important component, which is the subject of today's webinar. So, going back to the rat amygdala, now I'm going to talk about the role in cue-based appetitive pathway. So what I show you here is the central amygdala. If you inactivate the central amygdala in an animal that has already learned to recognize cocaine-associated objects or cocaine-associated place. So what happens is, if you inject an animal with cocaine repeatedly for 7 days, and then you do not give it any cocaine, you wait for 14 days, and then you put that animal back into that same environment, it starts showing more locomotion. This is what happens even in drug addicts, right? If you see the drug addict, if you put them back in that environment where they took the drug, they feel the craving. They have the memory of taking the drug. So it forms a strong memory. So that is a memory that I tried to map over here. That's what happens in this case. So if you look at that particular memory and then you see if you inactivate the central amygdala, that in itself is sufficient to cause all the responses in the cue to go away. In other words, if you put the animal back in that same environment, it does not show any recognition. It does not show increased locomotion. On the other hand, if you lesion or inactivate the basolateral amygdala, that prevents the learning itself. So the animal fails to learn to associate anything with the cocaine taking or cocaine seeking. So this becomes very important. So this circuitry where basolateral amygdala is the presynaptic input and central amygdala is the postsynaptic input becomes important. And electrical, last but not the least, electrical stimulation of the basolateral amygdala is very, very important because once you do that to the presynaptic circuit in an inactivated basolateral amygdala, you can get the response back. So that is totally cool. So any kind of cue responding that was abolished can be reinitiated. So this circuitry I tried to map and see what happens when you manipulate phospholipase D. So again, refreshing back, if you take the central amygdala and the basolateral connection, NMDA, which is glutamate, and L-type voltage-gated calcium channels, which are important for signaling, these are recruited in strengthening this response. So my idea was, I know that dopamine is the motivational signal. In other words, dopamine motivates the animal to go seek the cocaine. So that definitely plays an important role. The metabotropic glutamate receptor is important for recall. So our group had established that D1-like receptors. So here I'm showing you an example of chemical LTP. So again, using the same profile of baseline FEPSP and timeline, here you see if you give a high-frequency stimulation in a saline animal, this is a saline control, which is the hollow circles, you see no change. If you give the SCH, which is a D1-like receptor antagonist, you apply to the slice, there is no change because there was no change in the potentiation. So there is no dopaminergic contributions here. However, if you look at the cocaine group of animals, you take the slices from that cocaine trained group, what you see is the cocaine control shows a nice, beautiful potentiation, okay, by high-frequency stimulation. This potentiation is diminished by application of the SCH along with the high-frequency stimulation. So this is a very nice way to show how you can use drugs preclinically to test its effects on different synaptic circuits. This becomes very important for its translational application. So now what did I do? I wanted to see, okay, now that we know dopamine plays a role, can I use just dopamine as the chemical LTP maker? So that's what I'll show you here. So if you take dopamine, raclopride blocks D2 like receptors. So this is specific for D1 like receptors. What you see here is that there is potentiation in the cocaine group of animals, okay, not in the saline group of animals. If you use the D1 like receptor antagonist, you see that in the cocaine group of animals, it completely abolishes this particular response. So this encouraged me to then ask the question, okay, whether this LTP can be done using things other than dopamine. Because remember, dopamine is activating a lot more receptors. And I want to look at specifically dopamine 1-like receptors, which are involved in the motivational signals in cocaine. So SKF is a D1-like receptor specific activator. And so it mimics the profile. See, the hollow triangles show you how in cocaine it is potentiated, but it's not potentiated in the saline group of animals. Now, when you apply, again, the D1-like receptor antagonist in the cocaine group of animals, the response is abolished. So this clearly indicates to you that there is a reproducible, robust chemical LTP that you can use to study the synapse. So now comes the final data slide that I'm going to show you. So here I want to show you what happens. Okay, this is a culmination of a lot of mechanisms. So let me show you what happens. Here, I am generating a long-term potentiation using SKF, which is by the D1-like receptor. So you can see that that is what happens in the cocaine control. Now, I take that and I apply the PCCG13, which is a mGluR-linked phospholipase D inhibitor. If I apply that 5 minutes before application of SKF and continue through that time, and then look at the response, you see what happens to the colored triangles over here. Basically, the LTP is completely abolished. It is not even generated. This suggests that dopamine, which may be located upstream, is interacting with mGluR through phospholipase D. So this is the first time where we showed that there is convergence of two different neurotransmitter systems through phospholipase D in the long-term memory expression of cocaine responses or an appetitive memory. So this was the take-home using these studies. So to summarize what I've tried to show you so far, that you can do a mechanism mapping using just electrophysiology, which you can then corroborate, which I did. What are the advantages? You can do electrical stimulation, you can do chemical stimulation, you can look at slow responses, which are the field responses over a long period of time, an hour, 2 hours, and so on. So you can see long-term changes in memory, and the synaptic circuitry within a brain region is important. Now, I showed you two different circuitries, the thalamic to the LA and the basolateral to the central. There are also these different trisynaptic circuits in the hippocampus, and there are circuits between the amygdala and the hippocampus. So imagine, for us to completely understand the brain, which is not a homogeneous structure, we will need to map all these. So a combination of different electrophysiological techniques, genetics in invertebrates, is going to provide us a lot of information to do these studies, because mechanisms in brain circuits and synapses are key for developing drugs. We have had a lot of neuroscience drugs fail, and in fact, all of the companies in the past 10 years have shut down. All biotech companies making any kind of neuroscience drugs have stopped making them because the number of side effects are too many. This is because there is no specificity, okay? You can increase the specificity by understanding the mechanism, the regional signaling, like I showed you, and then we can try to look at therapeutics that will target these places. So with that, I thank you very much for your time, and I appreciate the presence. I'll be happy to answer any questions that I can.
Okay, thank you, Balaji, for that excellent presentation. And now we'll move into a few questions. So our first question is, Balaji, what are the advantages of field patch versus patch clamp?
Well, like I mentioned towards the end, field patch, I mean, if you look at the field recordings, you can map different circuits. As you can see, the brain is not a homogeneous structure. You require understanding of these different regions. In some places, NMDA is activated. In some places, NMDA is diminished. So if you give a drug that is going to increase NMDA levels overall, this is not going to be of any use at all. Using patch, you can look at individual neurons and how they respond, or individual synapses between two different neurons and see how those respond. But if you want to look at a population, the advantage is for the field studies, because that will give us more information. But this is there is a different way to look at it. Like I pointed out in this presentation, it is important to integrate different approaches. So I would say that the best way to do this is you ask a specific question and pick out the technique that will answer the question in the best manner. Is it field? Is it patch? Or is it something else?
Okay. Next question. Do you use optogenetic approaches to modulate different synaptic input on the basolateral neuron?
Not yet, but that is definitely one of the things in the work, because that is a powerful tool, and I would like to use it in combination with in vivo recordings also, because that is going to be the most efficient way to use the optogenetics.
What software do you use to analyze the data? And did you measure the max slope of field EPSPs?
Yes, well, I don't use slope, but like I mentioned, I do use the 30% of the maximum input-output that I get. So when I do these recordings, I do an input-output curve. I look at different current stimulations and pick out the 30% response, because when we want to look at excitatory responses, we want to pick the lowest possible recording that will give you a nice measurable slope. And
okay.
What was the first part of the question? I'm sorry.
So what software did you use to analyze the data?
I've always used ClampX. ClampFit rather. I found it to be most efficient. I know people use some of the other ones, but I found that I could do much more things using ClampX.
Okay. Can you do patch and field recordings at the same time on the same prep?
Oh yeah, that is why I got the 700B, because that is one of the best things to do. Because if you want to map a circuit, you need to have the facility to do both patch. So you're picking a particular cell in the postsynaptic area, and then you're also having another electrode right next to it, taking the field response. That is the best way to look at things.
So what are the advantages in recording from the amygdala versus the commonly recorded hippocampal synapses?
So advantages, I cannot say that there is an advantage per se. It is almost how easy it is technically. So when things were started off, of course, they started using young animals because it was very difficult to get any kind of recordings from older animals, where the synaptic circuits were more or less were not as amenable. The hippocampus, because it undergoes a lot more neurogenesis, you could record from that much easily. Plus, there are more defined synaptic pathways. The cellular layer is one particular layer. The fibers are in one particular area. So it becomes easy to map the circuits there. So it has been the one that has been extensively studied. However, like I pointed out, amygdala also plays an equally important, if not a more important role than the hippocampus. There are more connections that come to the amygdala than to the hippocampus. So you have connections that come from practically every sensory system, which parallelly goes to the amygdala, as well as the thalamus, which is then routed to the rest part of the brain. So amygdala plays a very important role in the fight or flight response. Even in neurodegenerative states, amygdala is one of the first structures to be affected. Sense of smell is one of the first structures to be affected. So, and of course, aggression is one of the first things that you see as a part of Alzheimer's disease, one of the first features. So amygdala as a brain region is very, very important to study. However, it's very difficult to study because, like I mentioned, it is very fibrous. It is very difficult to manipulate and navigate it. But once you get the hang of it, then there are a lot of things to be discovered there.
Do you have any behavioral data supporting the EPHIS data?
Yes. For each of the papers that I showed you, in everything I have behavioral data. And I have cocaine place preference. I have cocaine conditioned responses. I have fear conditioning responses. Yes. For each one of them, I have done pharmacological manipulation. So I direct the attention of the person to my papers.
What are the benefits of high-frequency stimulation LTP versus over the chemical LTP that you utilized in your studies?
Like I said, high-frequency stimulation gives you a powerful response. So, in other words, you're activating all the different signaling pathways. I mean, there is no specificity there. So this can be used to actually map out whether something has an all or none response, or is the gradation of responses of everything that is functioning. But if you want to ask specific questions, okay, is this NMDA dependent? Is this NMDA independent? Does this involve NGLURs? Is this an early or a late LTP? All of these can be mapped using chemical LTP. So you use specific agonists to activate different signaling circuits.
Okay, and last question. How could you make electrophysiological studies, such as the ones that you've done, more translational?
So, like I mentioned, I mean, you would have to, first of all, develop these circuits. You have to understand the mechanisms. That is the first step, and we are a long way away from that in trying to completely understand synaptic mechanisms. Once we understand the synaptic mechanisms, then it will be possible for us to target the pathways. And then you develop small molecule inhibitors or any other kind of manipulable agents that you can test preclinically, like I showed you in the synapse. And then you can see in pilot clinical trials how these drugs fare. So that would be a sequential development.
Okay. All right. Well, that concludes our webinar today. Thank you, Dr. Krishnan, for joining us and for giving that excellent presentation. And thank you to all of you for joining us. Last, I'll leave you with a promo that we're running right now. If you're interested in getting a new digitizer, we're currently running a promo until the end of March. That if you want to get a Digidata 1550B, you can get 4 channels of hum silencer for the price of the version that has no hum silencer. So it's an excellent savings. So thank you for joining us, and we'll conclude. Have an excellent day.
Thank you.